Imagine the thrill of potentially unraveling the mystery of ancient life beyond Earth. NASA has made a claim that could change the course of our understanding of extraterrestrial existence with its astonishing announcement regarding possible signs of ancient life on Mars. In a groundbreaking investigation centered on the Sapphire Canyon mudstone core, drilled by NASA's Perseverance rover in July 2024, researchers have found intriguing evidence that supports the long-standing quest to determine if life once thrived on the Martian surface. This study reveals minerals and textures that, if found on Earth, are typically associated with microbial activity. However, the research team also reminds us that there may be unknown non-biological processes that could account for these signals as well. This finding by Perseverance is the closest we have ever come to discovering life on Mars. The identification of a potential biosignature on the red planet is a groundbreaking discovery, and one that will advance our understanding of Mars, stated Acting NASA Administrator Sean Duffy, emphasizing the significance of this research and its implications for planetary science. But here's where it gets controversial. Many scientists remain cautious, alerting the public that this evidence is yet to be conclusively linked to ancient Martian life. The core sample was extracted from a rock referred to as Chavaya Falls, located in Neretva Vallis, which is an ancient river channel approximately a quarter-mile wide that previously nourished the lake in Jezero Crater. After the drilling process, the Perseverance rover meticulously sealed the sample for a potential return to Earth. This would allow state-of-the-art laboratory technologies to conduct tests that surpass what the rover can perform in its current Martian setting. When examining this Martian mudstone, lead author Joel A. Hurowitz from Stony Brook University observed a fine-grained mudstone featuring circular reaction fronts, informally known as leopard spots, alongside tiny nodules within the layered sediments. Within the samples, Perseverance's advanced Sherlock and Pixel instruments successfully charted organic carbon alongside phosphate, iron, and sulfur, arranged in distinct and repetitive patterns. Two key minerals of interest are vivianite and greigite. Vivianite, an iron phosphate, and greigite, an iron sulfide, are often linked to iron and sulfur cycling in environments that lack oxygen. Notably, these minerals typically appear in sedimentary rocks formed in aqueous conditions, rather than in volcanic lavas. The area where the core was collected reveals sedimentary features that suggest gradual changes after the mud was deposited, rather than the result of high-temperature formations. The significance of these findings cannot be overstated, as the temperatures in which these reactions occurred align with environments that life can thrive in, as opposed to extreme heat that would eliminate delicate indicators of biological activity. This detail shines a hopeful light on the potential for Martian life. Interestingly, similar microbial processes on Earth often yield the same traces. On our planet, vivianite forms under conditions where microbes reduce iron in waterlogged sediments, efficiently trapping phosphorus within blue-green nodules. Extensive laboratory and field studies document vivianite formation facilitated by biological processes, showcasing electron transfer mechanisms. Greigite, on the other hand, tends to manifest in environments dominated by sulfate-reducing bacteria that drive chemical reactions in oxygen-depleted areas, and experiments have shown that greigite only emerges in situations where biological activity is present. Martian rocks formations exhibit vivianite-rich rinns encircling cores abundant in greigite, a pattern reminiscent of the electron transfer processes noted in certain Earth sediment samples. However, it's crucial to point out that these findings do not definitively prove that metabolic processes occurred within the Bright Angel mud. They indicate that the environmental chemistry is conducive to supporting life. This distinction is essential, which is why researchers exercise caution in their language, avoiding undue speculation. The existence of a potential biosignature is fascinating. It refers to features that might hint at a biological origin, yet still require further data to rule out non-biological explanations. NASA advocates for a systematic approach through the Confidence of Life Detection, COLD scale, which champions meticulous evaluations to confirm findings. The initial step involves identifying a signal, then eliminating contamination factors, and subsequently probing alternative explanations before confidently addressing the possibility of life on Mars. The research surrounding Bright Angel is in the early stages of this investigative ladder, successfully completing several crucial steps while still necessitating rigorous testing in laboratory conditions. It's also important to acknowledge that organic compounds can reach Mars through meteorites or can arise through abiotic processes. The research team acknowledges these pathways and articulates how forthcoming analyses will further dissect these origins. The Martian rover's findings of organic carbon in multiple areas of Bright Angel reveal a fascinating geological history. Reaction fronts rich in vivianite surrounding cores that contain a higher concentration of greigite present a scenario consistent with iron and sulfur cycles highlighted in the paper. The mineral veins showcase calcium sulfate, while the mudstone displays a fine-grained composition and low levels of magnesium and manganese. Significantly, there's no evidence of extreme heating that would have disrupted the rock or rearranged its texture. The examined outcrop resides within layers of sediment formed by water traversing Neretva Vallis, a channel that once exhibited a steady flow into Jezero's ancient lake. While the low-temperature context leans towards life-sustaining chemistry, it doesn't necessarily signify the presence of life. Non-biological mechanisms can sometimes replicate similar patterns, which creates a tapestry of complexity regarding the planet's history and the potential for life. It's crucial to remember, however, that excitement must be tempered with sobriety. Nikki Fox, an associate administrator for NASA's Science Mission Directorate, cautioned that while these findings hint at a potential biosignature, they do not constitute direct evidence of life. Hurwitz echoed these sentiments, reiterating that claiming these findings as more than a potential biosignature would be premature. Furthermore, Sean Duffy, the acting NASA administrator, remarked that this discovery may indeed represent the clearest indication of life that we have yet encountered on Mars, stirring a mixture of hope and caution in the scientific community. This careful stance is not merely a hedge against overly optimistic claims, but rather a commitment to scientific integrity, especially when addressing such monumental questions about our place in the universe. Delving deeper into the implications of these findings shows that if vivianite and greigite indeed formed through microbial-like processes, the Bright Angel sample encapsulates a time when surface waters on Mars supported similar chemical mechanisms as some living organisms utilize for energy today. This discovery pushes the boundaries of Martian habitability into a period when this particular area of Jezero remained wet. Conversely, even as these mineral formations arose through abiotic means, they still provide vital insights into the redox organization of iron, sulfur, and phosphorus in Martian sediments, a narrative of how the planet's chemical landscape evolved through time. Such insights are critical to understanding Mars's climate trajectory. It didn't merely dry up, but underwent significant chemical transformations. The implications of this research also pave the way for future investigations. Scientists are now looking into isotopes, microtextures, and the precise composition of carbon within the core, which could help differentiate between biological signatures and their chemical counterparts. As the quest for life on Mars progresses, the authors put forth laboratory experiments and field analog studies to determine whether non-biological processes can replicate these specific textures and mineral configurations. They also advocate for analyses necessitating the sample to be handled in a pristine laboratory on Earth, including measuring isotope ratios that biology tends to influence. The planning of sample returns will significantly impact the pace at which these vital tests can be conducted. While Perseverance continues its mission to chart out the distribution of these geological features and their relation to neighboring rock formations, equipped with their sensitive instruments, Pixel and Sherlock have provided sufficient data to guide future exploratory efforts. This synergy of elemental mapping and Raman spectroscopy offers a coherent picture that researchers can apply to examine other significant Martian outcrops. As the investigation unfolds and new targets are cataloged, the cold framework ensures that the excitement surrounding potential biosignatures progresses in a methodical manner, maintaining trust in the scientific process. Ultimately, the study published in the journal Nature raises questions that beckon discussion. Do you believe these findings could indicate ancient life? Or are we too eager to find something that may not be there? What does this mean for future Mars explorations? Join the conversation and share your thoughts.